We're going to talk about one of the most important processes in biology, protein synthesis. This happens in two major phases, transcription and translation. And they together turn the information stored in DNA into functional proteins that keep cells alive. Let's start at the beginning, inside the nucleus with DNA. A gene is a specific sequence of DNA nucleotides that hold the instructions to make a particular polypeptide. DNA itself is a double-stranded, antiparallel molecule made of 4 nucleotides: adenine, thymine, cytosine, and guanine, which store all of the cell's genetic information and are held together by a hydrogen bond. Transcription begins when RNA polymerase binds to a section of DNA called the promoter. This signals where a gene starts. RNA polymerase unwinds the DNA and separates the two strands by forming a transcription bubble, breaking hydrogen bonds between the nucleotides. RNA polymerase builds the RNA strand using the antisense strand as the template. The enzyme matches the complementary RNA nucleotides. Adenine pairs with uracil, which replaces thymine. Thymine pairs with adenine. Cytosine pairs with guanine, and guanine pairs with cytosine. Because of these rules, the newly made mRNA is nearly identical to the sense strand of the gene, just with uracil instead of thymine. RNA polymerase moves along the DNA. A sugar phosphate backbone forms, which stabilizes the single-stranded mRNA molecule. Hydrogen bonds between DNA and RNA break, which free the mRNA.
If transcription is like copying a text, then translation is like reading that text to build something new. In cells, that something new is a protein. Translation happens in the cytoplasm on a structure called the ribosome, made up of proteins and rRNA. The ribosome has two main parts, the small subunit, which binds the mRNA, and the large subunit, which contains the catalytic center that forms peptide bonds. Inside the ribosome, there are 3 binding spots known as the A, P, and E sites. The A site accepts incoming charged tRNA, the P site holds the growing polypeptide, and the E site is where the empty tRNA exits. Translation begins at the start codon, AUG, which codes for methionine. This is a signal that tells the ribosome, begin building a protein here. Each 3 base sequence on an mRNA is a codon. Each codon corresponds to a specific amino acid. In our mRNA, there's actually a mutation, and there's another start codon. The matching molecule that brings these amino acids is the tRNA. Every tRNA carries an anticodon that binds to a specific codon and a matching amino acid attached by an enzyme called the amino acyl tRNA synthase. Initiation is the first stage of translation. In initiation, the small ribosomal subunit binds the mRNA. The first tRNA carrying the methionine pairs with the start codon. Then the large subunit attaches, forming a complete ribosome. Then there's elongation. In elongation, a charged tRNA enters the A site. The ribosome's peptide transferase activity forms a peptide bond between amino acids. The ribosome shifts, moving the growing chain into the P site. The empty tRNA exits through the E site. The chain repeats like a molecular assembly line, codon by codon, extending the polypeptide chain. You can imagine the ribosome as a printer, the tRNA as the document, the tRNA as the printhead, and the amino acids as the ink. Finally, there's termination. In termination, when a stop codon, the UAA, UAG, or the UGA enters the A site, no tRNA matches it. Instead, release factors bind, causing the ribosome to fall apart and release the newly made polypeptide. This chain will fold into a functional protein. And that's protein synthesis, a highly coordinated process that brings DNA instructions to life. From transcription in the nucleus to translation on the ribosome, every step ensures the correct protein is built, one amino acid at a time.